Okay, welcome everyone. This educational session is not intended for the purpose of providing medical advice. All information, content, and material is for informational purposes only and are not intended to serve as a substitute for the consultation, diagnosis, and/or medical treatment of a qualified physician or healthcare provider. The information supplied through webinars or on our webpage or by any representative or agent of the MSA Coalition, whether by telephone, email, letter, or other form of communication, is for informational purposes only and does not constitute medical, legal, or other professional advice. Good day to everyone. Today, the Multiple System Atrophy Coalition is bringing you the fourth of a new webinar series. The title of this series is From Bench to Bedside, the Promise and Hope of MSA Research. This series will highlight researchers who applied for and won a grant from the Coalition Seed Grant Program. That program, begun in 2013, has resulted in 56 MSA-focused research projects for a total of $2.85 million. Grant proposals addressing any of four distinct diseases, categories, pathogenesis, diagnostic markers, preclinical and clinical, are submitted each year. The proposals go through a rigorous review process by MSA researchers who volunteer their time to support this work. The finalists are approved by the Coalition's Scientific Advisory Board and ultimately by the MSA Coalition Board of Directors. Everyone in this process is dedicated to finding a treatment or cure for this disease. We all have skin in the game. Let's introduce everyone who will be a part of this webinar today. We'll start with the researcher whose work will be the focus of today's webinar. So Dr. Roy Freeman is a professor of neurology at the Harvard Medical School and director of the Center for Autonomic and Peripheral Nerve Disorders in the Department of Neurology at Beth Deaconess Medical Center in Boston, Massachusetts. Dr. Freeman is past president of the American Autonomic Society, past chairman of the World Federation of Neurology Research Group on the Autonomic Nervous System, and past chairman of the Autonomic section of the American Academy of Neurology. Dr. Freeman is editor of Autonomic Neuroscience, Basic and Clinical, and on the editorial board of the Clinical Autonomic Research and Journal of the Peripheral Nervous System. His research and clinical interests are the physiology and pathophysiology of the small nerve fibers and the autonomic nervous system. His research encompasses the neurological complications of diabetes, neuropathic pain, the autonomic complications of neurodegenerative disease, such as Parkinson's disease and multiple system atrophy, biomarker development and neurological diseases, and the diagnosis and treatment of autonomic and peripheral nervous system disorders. He has around 300 publications. He has been principal investigator on many neuropathic pain clinical trials and has been principal investigator on National Institutes of Health funded studies on the neurological complications of diabetes, on cutaneous biomarkers and neurodegenerative disease, and the neurobiology of stress. Two members of the MSA community have had the opportunity to put together this webinar series. Before going any further, I'd like to introduce the two of us. My name is Ariana Pitaro. I do clinical research in the Corona Lab at Brigham and Women's Hospital in Boston. And Mr. Larry Kellerman.
Yes, hi, I'm Larry Kellerman. I'm a board member of the MSA Coalition. I've been here for about 8 years, and between the two of us, we've put this together the last two times. I'm looking forward to talk with Dr. Freeman and with Ariana and you in the audience. Thanks, Ariana.
Thank you, Larry. So the grant we'll focus on today falls in the category of diagnostic markers. Currently, there is no diagnostic marker for MSA, which is vitally needed to contribute to the diagnosis, treatment, and disease modification of MSA. More information can be found at multiplesystemaatrophy.org. Dr. Freeman applied for an MSA Coalition Seed Grant in 2013 in the amount of $25,000 with the project titled Biomarker Development in MSA. Today, we'll get to hear his story about the research. Welcome, Dr. Freeman, and thank you for taking the time to share your story.
Thanks very much. I think that's the first time anybody has introduced me and pronounced the word autonomic correctly. So this really is a record. It's been pronounced so many different ways: automatic, autoimmune. So that was great. I got it was a pleasure listening to the pronunciation. So let me share my screen. Thank you for having me. I know that most of you listening now and sometime in the future, and I always find that a little eerie, are patients or caregivers, caretakers. I want you, I'm going to I also know that many of you know more about multiple system atrophy than the average neurologist. So I'm going to address the talk to a knowledgeable audience. If there's something, well, first of all, if it's, if you find it boring and you know it all, please mention it in the chat. I'm thick-skinned and have an enormous ego, so it's not going to bother me. And if it is, if the areas that you want some clarification, please do the same in the chat. I may not be looking at the chat all the time, so Larry and Ariana, if you can just let me know that somebody's asked a question, and I don't mind being interrupted, at least,
Sure will.
Not too much anyway. Okay, let's share the screen. And here it is. How's that? Can you see the screen?
Yes.
Yeah, good. All right. So, I forget what the grant was entitled. It was, as Ariana mentioned, quite a while ago. And I will be talking about a biomarker for multiple system atrophy, and it's going to be a very narrow talk. I'm not going to talk about biomarkers in general, and there are a couple in play. I'm going to talk about a cutaneous biomarker, which is to say a biomarker in the skin. I do have a few disclosures which are listed over there. And one important disclosure that I have to make at the outset is that there is a manuscript based on this work that is in the very, 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 very late stages of review in a journal. And the journal has a very strict embargo policy. So I'm only going to be talking about material that is in the public domain, which is to say material that I've presented at academic meetings in the past, and hopefully in the next, I'm hoping, days, I will be able to release the manuscript. I mentioned cutaneous, and cutaneous means skin, and I'm going to go back a little in history and talk about skin biopsy, because if you are thinking, what has the skin got to do with multiple system atrophy? It's a very reasonable question. And so I'll begin by talking about what is a skin biopsy. And in order to understand that, you need to know a little bit about the skin. And the skin, as you know or may not know, is comprised of a superficial layer called the epidermis, which is a number of subcomponents, and then the dermis. And a lot of the action happens in the dermis, but the epidermis, and I won't go into it right now, is extraordinarily interesting, is in fact quite fascinating, even though it looks like it's just a shield on the surface of the dermis. And we do a skin biopsy, something like that, and we grab a piece of skin going through the epidermis into the dermis, and that allows us to look at all the beautiful and wondrous structures we're going to talk about. Now, I know, when we talk about a biopsy, it sounds like you're doing a heart transplant. It really isn't. It's a tiny little hole. That's a quarter. So it gives you some perspective of the size of the biopsy. For those of you who are European, here's a euro. It hasn't changed in size. It's pretty much the same size, tiny. We use sterile technique, but no with topical anesthesia, no general anesthesia, no sutures, and you put a Band-Aid over it and go home and don't shower for a day or two. And there is, we have not had a complication. Some some centers have reported occasional complications, but by complications, continuing bleeding, little infection, we have and have done thousands, tens of thousands of these have never had a complication. So it's really, as they say, a very benign procedure. So the next step is, okay, that's the skin biopsy. What about skin biopsy as a diagnostic tool? And I showed you the structures in the skin. What I didn't show are the nerve fibers. And there are nerve fibers in the, and I know this is going to be really difficult to see on your screen. It's difficult to see not in the dark, but in the epidermis, there are a number of epidermal nerves. I don't know, Larry and Ariana, can you see where I'm pointing? Does this project? Yeah.
Yes.
So these are the intraepidermal nerve fibers, and these come from branches in the dermis, and these are stained with an immunostain called PGP 9.5 protein gene product 9.5. And when you have a peripheral neuropathy, these are lost in the skin. And in fact, this is something that really has developed, I don't know, in the last 30 years. There used to be a neurological disorder called a small fiber neuropathy. And the small fibers were, we often use the term, invisible, because there was no way to diagnose these lack of small fibers, damaged small fibers, loss of small fibers, and the skin biopsy allowed us to do it. And this was a landmark paper, one of two landmark papers in the very late 1990s, in which the concept was introduced of the diagnosis of small fiber neuropathies using the skin biopsy. And now it is widespread, widely used by neurologists to diagnose this actually very, very common disorder, small fiber neuropathy. Now, we, as you know, my lab is interested in the autonomic nervous system, and we extended those studies looking at the intraepidermal sensory nerves to begin to look below the dermis. There you see the intraepidermal sensory nerves, but we began to look below in the dermis at the autonomic structures in the dermis. And these include the innervation of sweat glands, the innervation of blood vessels, the innervation of piloerector muscles. And there is a whole window on the autonomic nervous system: pseudomotor, vasomotor, and visceromotor. And visceromotor means viscous, like the gastrointestinal tract or the bladder, but the piloerector muscles, those muscles that stand on end when you are scared or stimulated, hear a beautiful piece of music which gives you goosebumps, those visceromotor fibers, those piloerector muscles kick into action. And these, in autonomic neurodegenerative diseases, are sometimes lost. And that's a long story, unrelated to muscles, MSA, or not unrelated to it, actually part of MSA, but unrelated to the talk I'm going to give, but is critically implicated, as you will see. And discovering how to measure and uncover these autonomic structures in the skin was a critical building block. And we published several times in the early mid-2000s, 2009, 10, 11, as to how to and what the implications are of looking at these autonomic structures in the dermis. And here you see a cartoon of it, the piloerector muscle, which gives you goosebumps when you see a love object, hear a beautiful piece of music, the blood vessels, which allow you to vasoconstrict when it's cold or dilate when you are blushing, and the sweat glands. We'll come back to why these are so important, but I just want to show how beautiful the pictures are. This is the sweat gland. This is with a marker, VIP, which is a sympathetic cholinergic marker showing the innervation of the sweat gland, which is shown in green, so red are the nerves. And it was a rather complicated study to quantify how you measure this. It really is like a study to attempt to measure how much spaghetti there is in a bowl of spaghetti. And we did this, I think, in a very beautiful and creative way, but the pictures are just very, very pretty. Here's the innervation of a blood vessel, and you see in red the sympathetic innervation of that blood vessel. Green is the blood vessel.
Wow.
And here you see that piloerector muscle, which, as you can tell, I'm very fond of, with its innervation beautifully shown in red with a sympathetic adrenergic marker, tyrosine hydroxylase, and those are the muscles that give you goosebumps.
Wow.
Yeah, it's, so really just quite beautiful. All right, let's begin now, having introduced the skin biopsy and introduced the structures that lie within the epidermis and dermis. Let's begin to talk about alpha-synuclein. And the story began, many years ago, when it was recognized that Parkinson's disease is actually not just a disease of the central nervous system, but also is a disease of the peripheral nervous system. And I and a couple of others around the world began to think, well, why don't we look for alpha-synuclein in the skin? We can characterize these structures in the skin so beautifully. Why don't we do that? And pretty much at the same time, a number of us began to investigate this. And the reason for investigating, in large part, was there was a critical need for a diagnostic biomarker in Parkinson's disease. Now, to take a step back just for a second, I know many of you who have multiple system atrophy or have friends or family members who have multiple system atrophy recognize just how long it took to make a diagnosis of the disease. And you probably think, well, patients with Parkinson's disease have it easy. It's so easy to diagnose Parkinson's disease. Physicians know about it. Everybody's heard about it. Nobody's heard about multiple system atrophy. Well, the truth of the matter is that it's not so easy to diagnose by Parkinson's disease either. And there was, in the early 2000s, a series of articles summarized in this one by Beach and Adler, showing just how challenging it is and continues to be to make a diagnosis of Parkinson's disease. And when we initially began our research, it really was to develop a diagnostic biomarker for Parkinson's disease. And we thought, well, that is, there's this, look where the light is shining. We know Parkinson's disease is a disease in addition to the central nervous system, but also of the peripheral nervous system. Let's look in Parkinson's disease first. But here I want to show you the diagnostic inaccuracy, and this is an autopsy study, was really pretty low, 38%, 65%, 53% of patients who were clinically diagnosed with Parkinson's disease were confirmed to have it at autopsy. So Parkinson's disease also is frequently misdiagnosed. So an average diagnostic accuracy of only 58%, which is really low for a disease that every medical student is taught about. Anyway, we thought, okay, let's address this and begin to see whether we can find alpha-synuclein in the skin and use that as a diagnostic marker. And we did, and here you see it. I don't know, on my screen, you guys are blocking it. So let me move Larry and Ariana and the MSA Coalition logo out of the way so I can point and show you. Here you see, and I want you to get used to this because this is going to be the theme, PGP 9.5 in green, and that's the panaxonal marker. The panaxonal marker, which is to say the one that stains the nerves, the intraepidermal and dermal nerves. And here in red,
Roy,
yeah,
before you go on, we have an audience member who has raised his hand, would like to ask you a question.
okay, go for it. Yeah.
Leonard, if you are ready to ask your question, I see you've got your hand raised. You want to ask the question, Dr. Freeman? Let's see.
Yeah.
Two participants have raised hand.
Okay, you will. Yeah, I
I
know. I see you are one of them, Larry. If people.
Well, I don't want mine up.
Okay, so we're down to one. Leonard, if you're up, Leonard, that's good. Otherwise, you can put it in the chat, or should I keep going?
I would say yes. If he wasn't, go ahead and put it in the chat, Leonard, if you would like to, and we can go from there. Okay.
Okay. And, you know, by the way, the two of you, feel free if there's a conceptual lack of clarity, please let me know to make something clearer. So theme is green PGP 9.5, staining the nerves, the axons in the nerves. Panaxonal means the nerves. And red is an immunostain for alpha-synuclein. And what we're interested in, because alpha-synuclein, as Ariana probably knows because she works in a lab, there's some immunostains which are sticky. They stay in the background. They make it extremely challenging to know whether you are just looking at an artifact, something in the background, or something real. And I will show you in a second how we get around that. But green, the innervation of the structures, you begin to recognize, doesn't this look like a sweat gland? This you can see, these two are piloerector muscles, and there you see the alpha-synuclein in red. Well, my slide isn't moving forward. There we go. All right. And we showed that in a subsequent paper, this time in 2016, the first one was in 2013, that across the array of Parkinson's disease patients, early, late, with autonomic, without autonomic dysfunction, we could see alpha-synuclein, and it was a sensitive and specific diagnostic biomarker in patients even without autonomic failure. And this was setting the stage for, and I'm sure this was the preliminary data that we gave to the MSA coalition, which encouraged them to fund this work. Here I want to show you we have PGP and alpha-synuclein. This is a sweat gland. You probably know another sweat gland, another sweat gland. Here you have the piloerector muscles, but importantly, here's the epidermis. And although we see nerves in the epidermis, we don't see any alpha-synuclein. So those sensory nerves don't have alpha-synuclein in them. Right, and this is what happens when we do a merged image, because the important thing conceptually is to know that it's not background, that it actually is within the nerve. And when it's golden in that particular plane, that means we are looking, and I'll take you through this one at the bottom right-hand corner. That means we are looking at real alpha-synuclein deposited in the nerves. And here is another image just to show you how pretty it can. Whoop, I didn't want to do that. You know, I wanted to do that just to show you how pretty it can be. And you can see it in 3D rotating around. The alpha-synuclein alone over here, and the golden is when it merges. It merges with using a computer. This is not a recipe where you mix it all up, but it's a computerized mersion where gold is a positive readout. Okay, now let's move to multiple system atrophy, and in the interests of time, I know you know all of this, and I decided I wasn't going to talk about the disease itself, and I wasn't going to talk about what's happening with other biomarkers. I was just going to focus on the skin. But if you have questions with regard to the disease, questions with regard to other biomarkers, feel free to put them in the chat or to email me.
Roy?
Yeah.
Where we are right now, I have a question for you.
Sure.
We're told also when we're studying that alpha-synuclein is here whether a person has a disease or not. It's in the blood, or it's in the brain. Alpha-synuclein is there. And so what made you guys think that, well, alpha-synuclein in the skin means disease?
Okay, so that is such a good question. And I decided to make this talk simple, which is always a problem when you've got smart people in the audience. So there is normal alpha-synuclein, which we all have, and then there is pathological alpha-synuclein. And the pathological alpha-synuclein is characteristically phosphorylated, usually at various sites along the alpha-synuclein molecule. At serine 129 is the characteristic site, and that phosphorylation encourages misfolding and aggregation. And that is the pathological alpha-synuclein. Now, in early studies, we looked at both normal and phosphorylated and worked out ways, mathematical ways, to differentiate between the total alpha-synuclein, which was both phosphorylated and non-phosphorylated, and the normal alpha-synuclein, the native alpha-synuclein. In the study I'm going to show you and talk about more, I'm going to focus purely on the phosphorylated alpha-synuclein. But that's a really smart question, and I just, in the interest of simplicity, decided not to discuss that until you ask the question.
Thank you.
Okay, so not going to talk about the disease itself. There actually was a really lovely article, August 2022, all about multiple system atrophy, which I really do recommend reading in Nature Neuroscience. So now let me talk about the MSA Cutaneous Alpha-synuclein project, which, as you heard Larry say, was seed funded by the MSA Coalition. It became a rather big project, and we actually obtained funding from the NIH to continue, and we were able to enroll a large number of patients. In fact, the largest MSA group of patients who had skin biopsies, and also patients with Parkinson's disease and normal controls. And I'm going to present those data and the conclusions of that study. So just an overview. I mentioned earlier there is a critical need for a diagnostic marker in Parkinson's disease. It's even more important in multiple system atrophy. And I emphasize again, many patients with multiple system atrophy are misdiagnosed early on. I think there's not a single patient that I see that was not diagnosed with some other disease early on. Functional disease, making it up, doesn't make sense. Progressive supranuclear palsy, Parkinson's disease, and we recognize that we really need to have a simple, easily executed diagnostic tool. It's also become increasingly important now that there are clinical trials to modify the natural history of the disease, and we all want these drugs to work. And one of the problems is that if you are in, and I'm not going to go into the details of sample size estimation, disease stratification, but if you are enrolling the wrong patient, non-synucleinopathy patients in a clinical trial, the drug will not work because most of these drugs are narrowly targeted to synuclein. So it's really important that you make an accurate diagnosis in your clinical trial of patients who are entering the clinical trial, because you want to treat patients who have, at the very least, synucleinopathy and ideally multiple system atrophy. We also wondered and hoped that what we were doing would have some therapeutic consequence. Now, one of the approaches when you are developing a drug, say a pharmaceutical company, is to make sure that you are hitting the target. And the skin is a simple way, with synuclein being present in the skin, to making sure that you are actually hitting the synuclein, because you can't do a brain biopsy, but you certainly can do multiple, time and time again, skin biopsies. It's also possible that you could use the skin as a surrogate endpoint. You could show a decline in the presence of alpha-synuclein in the skin in a clinical trial. Again, a very easy way to do it, because as you know, at this point, it's very challenging to image synuclein in the brain because it's inside the cells, unlike Alzheimer's disease, where you actually can visualize the amyloid on a scan in the brain. And so, diagnostic biomarker, therapeutic biomarker. We also wondered whether it would be a prognostic biomarker. Would it be, is the case that high alpha-synuclein in the skin could progress, have a disease which progressed rapidly, whereas low alpha-synuclein would progress slowly? But for this first cut, this first piece of research, all we did was to look to see whether it was a diagnostic marker, nothing to do with clinical trials, nothing to do with target engagement, nothing to do with prognosis.
Roy, we have one question came in.
Good. Yep.
Joanne Govas would like to know, how common is small fiber neuropathy in MSA? Do you see MSA?
Okay. Well, a couple of points. Small fiber neuropathy is fairly common, period. Next, in MSA, although we initially thought it was completely the other side of the coin to Parkinson's disease, that's not the case. It is very clear that in patients who have MSA, that there is also loss of the small fibers and of the autonomic fibers, but it is not to the extent that we see in Parkinson's disease. And in fact, you have preempted the results of the study, because that was one of the features that we found, that there was definitely a decrease in enteroepidermal nerve fiber density in MSA patients, but not as much as in Parkinson's disease. Joanne, you cool with that answer?
We'll find out, hopefully.
Okay, all right.
Thank you.
Yeah, by the way, everybody, I mean, just interrupt me as this thing goes on. It will get a little bit more complicated, maybe interesting. So I'm hoping for interruptions. Okay, so I showed you how in Parkinson's disease that there's a critical need for a diagnostic biomarker, and I mentioned that the patient journey, as we say, is a rather circuitous, hazardous one in patients with MSA. Is this in fact true? Well, of course it's true. Autopsy studies have shown that of patients with clinically diagnosed MSA, only 60% had the correct diagnosis as autopsy. And the most common misdiagnosis was dementia with Lewy bodies, followed by a disorder known as progressive supranuclear palsy and Parkinson's disease. And this was a paper reported in 2015, an autopsy study looking at patients with clinically diagnosed multiple system atrophy. So it's really the other side. It's not talking about the patient journey. It's saying once patients are given a diagnosis of MSA, what did they have? And here you see that 62% accurate, that's the non-MSA diagnoses, Lewy body dementia, Parkinson's disease, progressive supranuclear palsy, and a bunch of other diagnoses. So these are the 38% misdiagnosed, and this adds up to 100%. Another study, that study was from the UK, from the US. This is a UK study. They entitled it "Improving the Diagnostic Accuracy of Multiple System Atrophy." They had some suggestions, but in that study, a little bit more accurate, 78% accurate, MSA, non-MSA, and Lewy body dementia again, progressive supranuclear palsy, cerebrovascular disease, ALS, cerebellar degeneration. So again, wherever you live, the diagnosis of MSA may be wrong on the one hand, or it may not, and likely is not diagnosed. Were this a live audience, I'll ask people, I would say, how many of you took 1 year, 2 years, 3 years, 4 years, 5 years to be diagnosed? So what I'm going to do now is I'm just going to ask, I know this is a group that's reluctant to put to ask the questions in the chat room, but I want you to put a number in, and Larry and Ariana are going to give me the numbers. So if it took 1 year to make the diagnosis, put 1 year, and you can say what the first diagnosis was. And at the end of the talk, or when we get a reasonable sample, Larry and Ariana will tell me what the distribution was and what the misdiagnoses were. So at the moment, I only see, I see nothing in the chat. So I don't want you to sit on your hands. There are 15 of you, so come on.
You see, got one response in the Q&A, still searching for diagnosis. So
okay, well, that's an interesting one. Good. All right. So, you know, as an aid to physicians, the group in the UK introduced red flags to differentiate multiple system atrophy from Parkinson's disease. I'm not going to go through these just in the interest of time, because I'm talking much more slowly than I thought I would, but there are a variety of clinical features that you see here that knowledgeable physicians may use. A critique of the consensus criteria that we used, by the way, in our study, because it began before this study was published, came up with a number of flaws, and you know, some of your physicians may be listed on this paper. So I was one of them, so I can say we came up with a number of flaws in those diagnostic criteria and created areas in this paper where we thought it was important to differentiate multiple system atrophy from Parkinson's disease, multiple system atrophy from dementia with Lewy body. This is another talk, a clinical talk, which I'll be very happy to talk in another symposium, but you've got some, you know, you've got some other very good clinicians who can talk about this. But essentially, you see the 4 points, MSA versus PD, 3 major points, MSA versus DLB, and we are getting better at the clinical differentiation. And came up with just this year, in fact, published just a month or two ago in press, a new set of diagnostic criteria, which you see here, which was an update on the previous consensus criteria. Now, in those, with those, we hope to do a better job. But despite this, I think everybody knows, every clinician knows, and I think every patient knows that we are not there yet, and the need for a diagnostic biomarker is critical. It is a serious unmet need. And specifically, I'm going to focus on increasing diagnostic certainty, and that can be used for clinical practice and in clinical trials. So The goal of the project, and this was really what initially, I didn't look up the proposal to the MSA coalition, but it was very much along these lines, because MSA and Parkinson's, particularly Parkinson's with autonomic failure, are so difficult to discriminate, even for the educated eye. We want to develop an accurate, accessible diagnostic biomarker, and we hypothesized that the cutaneous, Larry's point, phosphorylated distribution pattern, coupled with the assessment of intraepidermal nerve sensory fibers, Joanne's question, and the dermal autonomic nerve fiber density, nobody's asked that yet, and other clinical measures would provide a unique pathological signature. And to make that hypothesis a little clearer, we thought that MSA is pretty complicated. Multiple is multiple, which already implies complexity, that one single biomarker is not going to do it. And so we thought we would need a little complexity in the diagnostic approach in order to establish high sensitivity and specificity. We were only a little bit wrong. So as I said, high number of, look, it's not a lot of MSA patients in the greater scheme of things, but for a skin biopsy study, 31 patients is a lot, 54 with Parkinson's disease, 24 controls. We evaluated them with structured examinations, with the structured smell test, tends to be lost in Parkinson's disease and dementia with Lewy bodies, less so with MSA, autonomic testing, and it was collaborative, part of this NIH study, Boston, NYU, and Denver. And we did skin biopsies, 3 of them, at the distal leg, distal thigh, and behind the neck. And the reason we did that is because we were aware of various theories regarding the way alpha-synuclein is transmitted from the periphery to the central nervous system, from the central nervous system to the periphery. And we looked at all of those nerves, the sensory nerves mentioned by Joanne, the pseudomotor nerves, the pilomotor nerves. We looked at phosphorylated alpha-synuclein, and we used confocal imaging with optical sections at 3-micron intervals. So it's microns sound thick, but these 3 microns is actually thin. Microns sound thin, but 3 microns actually is thick, because we want to be sure that we are actually looking at nerves and alpha-synuclein. So it was critical to the way we do these studies that we do, that we look at a nerve that we can follow through the dermis. And what did we find? We found that phosphorylated alpha-synuclein was present in at least 1 biopsy in every patient with MSA, not quite as many as in Parkinson's disease, and in none of the patients, in none of the control subjects. So already we have a nice way to differentiate synucleinopathy patients. We didn't do dementia with Lewy bodies, but in other work we've shown very much the same thing. We didn't report, in this study, we didn't report pure autonomic failure, nor REM behavioral disorder, which is one of the precursors. Another study, we've shown that too, but this is just MSA versus PD, which is the major differentiation. Contrary to what we thought, phosphorylated alpha-synuclein was significantly higher in patients with MSA compared to Parkinson's disease at every diagnostic site. Not only that, and this was a fascinating knock you out of your chair finding, the topography of the deposition was different between MSA and PD. And I'll explain that in a little bit more detail. And then finally, the combination of a couple of these metrics yield sensitivities and specificities of above 95%, 97, 98%, which is incredibly high. So we are thrilled about the results of this study. Now, when I mentioned the topography, I'm going to have to take a step back and talk you through this. Now, in multiple system atrophy, there was a fairly diffuse distribution. This is PC is the back of the neck, the posterior cervical region. DT is the distal thigh, and DL is the distal leg, and 31 is the number of subjects that we have. So every time you see something here, it's a subject, and you see that the MSA patients had more, bigger the circle, more alpha-synuclein, and it was pretty evenly distributed. The Parkinson's patients shown over here, 54, every time you see a dot, it's a subject, had less and much more in the neck, and getting less than in the distal thigh, and even less in the distal leg. So 93 to 33 to 17 in the Parkinson's patients, what you see over here, whereas 84, 87, 77, very even in the leg. Let me show you that differently. So Parkinson's disease, 94, 93, 33, and 17, seen over here. MSA, pretty even, so a different distribution. The question that's going to be asked is why? And the answer I will give is I have no idea why, but we do know that the conformation of the physicochemical qualities of alpha-synuclein is different in MSA compared to Parkinson's disease, and it very likely plays a role in the brain deposition. Why is the deposition different in the brain? Why are different structures involved in the brain in PD and Lewy body dementia and MSA? And the same thing is very likely in the periphery. So that's the percent positive by disease by site. Here we see the topography. And I want to talk about an index that we used to measure the distribution. Now, if there was a rostral to caudal gradient, which to say the kind of gradient that we see in Parkinson's disease, rostral meaning the head to caudal in the distal leg, if there was a rostral to caudal gradient, you would have a +1. Whereas if you had a -1, it would go from the legs to the feet. And here we see Parkinson's disease, it's roughly 6.64, so getting close to the +1, whereas MSA, it's pretty even, it's almost hitting 0, so not caudal to rostral, but really pretty evenly distributed, as you saw in that slide. So this topography, for me, this blew me out of my chair when we looked at it. Now, what does this actually look like? Well, I'm just going to ask you to focus on the red over here, and on the red is MSA, red is alpha-synuclein, that's the neck, this is the distal thigh, this is the distal leg, lots of alpha-synuclein. Here in Parkinson's disease, lots in the neck, not too much in the distal thigh, very, very little in the leg. And here you see it. Remember I said red alpha-synuclein, green, the innervation of the structure we're looking at, and merged images on the side. So that's really what we look at and what we quantify. Now, when you have a diagnostic test, you really want to know how accurate it is, which is a measure of the sensitivity and specificity. And we found that there were a number of ways to measure, to accurately differentiate Parkinson's disease from multiple system atrophy. And these involved the amount of alpha-synuclein, the distribution, which is to say the topography, Joanne's nerve fibers, the presence of intraepidermal nerve fiber density or not, and a number of other areas. And I'm going to show you, rather than talk you through it, I'm going to show you just how good it was. So this is a measurement of the deposition, the amount. Black, MSA, white, clear, Parkinson's disease. This is the distribution. Remember I said 0 is pretty even. +1 is where a lot of the Parkinson's lie. this side is where the MSA lies. And if you combine these, you can create a very, very narrow you can create strong separation with a very, very narrow overlap between the diseases. And this was the best combination. So if you were to say, what's the best way to differentiate MSA from Parkinson's disease? I would say the combination of the amount of deposition and the topography of the deposition. But there were other areas that were really good too. This is the nerve fiber density. So if we look at the nerve fiber density, as I mentioned, Parkinson's disease, low intraepidermal nerve fiber density, multiple system atrophy, not normal. The population that Joanne asked about is over here, but also pretty good differentiation. Here we have, I'm showing you this in 2D, but we actually attempted to look at it in 3D. This is, we'd known for a long time that MSA patients have a better heart rate response, perhaps related to the better innovation. Reported that quite a while ago. Another way of differentiating, another way of differentiating, which one can do very, very nicely, a little bit of overlap, but very, very small. And then finally, looking at that smell test, again, Parkinson's disease, decreased sense of smell to a quite profound disease, multiple system atrophy, relatively preserved. Deposition, sense of smell. Now, so we have the frame. Now, these were patients that I felt really comfortable. These are physicians, my group, the NYU group, Colorado group. I trusted the diagnoses. I don't know if they were 100% accurate. This overlap that we saw in maybe the areas where we made mistakes, but this is pretty good. As we go out into the community and do more and more of these, I think the diagnostic accuracy may not be quite as good, and we may need to bring into play some complicated statistical approach using smell, using heart rate, using intraepidermal nerve fiber density, in addition to looking at deposition and the distribution. But this is the framework which we are going to use. So in conclusion, we can differentiate MSA patients from those with PD and healthy controls with excellent, really high sensitivity and specificity. This is, 96, 97, 98% is incredibly strong, using cutaneous neuropathological assessment and selected clinical features. As I said, it's present within cutaneous autonomic nerve fibers in patients with MSA and PD, but not in healthy controls. Greater deposition in MSA compared to PD. The topographic distribution varies. PD, proximal to distal, head greater than legs, whereas more evenly distributed in patients with MSA. Patients with PD have a mild length-dependent sensory neuropathy that it's not present in patients with MSA, or not present to the same extent, I would say. And taken together, we believe that this is strong support for skin biopsy as an accurate method to pathologically confirm a diagnosis. Now, when we, as I said, go out into the community, the clinical data, the autonomic data may be necessary to keep up this 96, 97, 98%, but at the moment, in patients who are what we can say that we have strong confidence, it's really, really good. So, conclusion, assessment of alpha-synuclein deposition within the skin has the potential to provide a safe, accessible, and repeatable marker of disease pathology. On the left are the collaborators in this specific study. On the right are people who've played a role in this all along the way. I mentioned the NIH because they funded a substantial aspect of this project, the Langer Family Foundation, of course, and the MSA Coalition. And I really want to thank very, very deeply the MSA Coalition for spearheading and getting this project going. That's it. Any questions? And do we have any sense of the distribution, how many years it took to make a diagnosis?
No, we only had those two. But we
Okay.
have some questions for you based upon what you just have given us that have just come in. So if you want to, we'll start with those. And then Ariana and I have a couple of questions we thought might help do a little clarification. We have a bit of time, but I want to start with the question from an anonymous attendee is, the skin test used the SIN1 test. And I think following that, another question or another person pointed out, this is fascinating since I just got my SIN1 results last week. So is the SIN1 test the one that is?
Okay, so, let me, as I want to make a couple of points very clear. As you saw in my disclosure, Cutaneous Diagnostic Company, which is the company that does the SIN1 tests, is one of my disclosures, and I am in fact one of the founders. In very brief, Chris Gibbons, my collaborator, and we were getting a lot of requests for doing this test in our lab, and we just didn't have the bandwidth to do it and didn't want to become a clinical lab, and so we explained to an existing lab how to do the test, and we are now scientific consultants, that's Chris Gibbons and me, to the company. Now, what the company does is a little different to what we do, and this MSA data that I spoke about is not part of what they do, but it may well, once it is published, may well become part of what they do. That is up to the company, and I'm not the company, I'm just a consultant to the company. So is this skin test what's used in the SIN1 test? What is in large part used in the SIN1 test is what I spoke about in the beginning when I spoke about Parkinson's disease and other synucleinopathies. The MSA differentiation from Parkinson's disease is not part of the SIN1 test.
Very good. Okay. Are there any other questions from individuals? If not, Ariana, I think you had one or two that you wanted to ask. And I have one. He's already answered the other one through the course of the conversation. So do you have one, Ariana?
Yeah. First of all, thank you so much, Dr. Freeman. This was very informative. Do you have an idea of, has there been any work done to determine kind of how early on in the disease course the test can, detect the phosphorylated alpha-synuclein in the skin?
Yeah, that's a really good question, of course. So what we do know is that in a substantial, well, maybe take a, just because, I appreciate you asking that question for the audience, maybe I should take a step back for the audience and say, one of the fascinating aspects of the synucleinopathies is that there are premotor, there are premotor features. Premotor features include loss of sense of smell, constipation, REM behavioral disorder, which is acting out dreams. Part of the nervous system degenerates. So when we have vivid dreams, usually these are not translated into the motor system. REM behavioral disorder is one of those disorders in which there's loss of that inhibition, so people act out their dreams. Now, with respect to these premotor features, we have shown, in fact, several groups, not just us, have shown that alpha-synuclein is present in patients who have REM behavioral disorder. What we've not shown yet is whether it is a predictive biomarker, a prognostic biomarker, how soon those patients will develop a synucleinopathy. But what we do know, the natural history of REM behavioral disorder is over the course of 10 years, a substantial number of patients will develop an alpha-synucleinopathy. So the quick answer to your question, fairly early on, very early on probably. And when I spoke about clinical trials and drug development, always you want to start using a drug that disease modifies as soon as possible. And I think the growing interest is in getting patients who have disease, true disease at the earliest stage. And it's hopeful that this kind of approach will help get patients into clinical trials and on treatments once drugs are approved as soon as possible. So it's a really important question, how early. We don't know exactly how early, but that gives a clue very early.
We have one more from an anonymous attendee. If the skin test comes back negative, is that a sign that you don't have MSA 100%? And the reason he asked, apparently it may be, but he's got in below, his mother was first diagnosed with MSA cerebellar about 18 months ago, based on some symptoms. And I guess that's what the question relates to.
Okay, so I want to take a gazillion steps back and talk about the diagnostic process. And when you see a patient as a clinician, it's a little bit like a puzzle. And what you are attempting to do is to come to a diagnosis, and you use a number of pieces, a number of bricks to build a diagnostic conclusion. And to do that, you take a history, you take a family history, you do an examination, you do a variety of tests, autonomic tests, neurophysiological tests, imaging studies, and a skin biopsy. And you don't need to do all of those. You don't necessarily, depending, the physician may not want to do imaging, may not want to do autonomic testing, may not want to do the skin biopsy, but it's a piece of a puzzle and you are building a diagnosis. So it's brick by brick by brick. So if one test, and the question was if the skin test comes back negative, if one test comes back negative, it's just one piece of that wall, that building that you are attempting to construct in order to come up with a definitive diagnosis, and no aspect of it, the history, the examination, the autonomic testing, the imaging, the skin biopsy, is something that is going to be an all or none. So I want to make this point because I think there are misconceptions out there with respect to tests in general, imaging tests, autonomic tests, and also the skin biopsy. So as always, you need a good and thoughtful doctor who's going to put all of the pieces together. And I think my view is the skin test is an important aspect of the diagnostic process, but it is not go, no go. It's not yes or no.
I thank you for that response, Dr. Freeman. That is very important, I think, for many patients to understand wherever they are in the disease diagnosis process right now. Ariana, do you have anything else that you need to ask, or should we be closing this? It's about time.
No, I think my questions have been answered. Thanks so much, Dr. Freeman.
Do you want me to do anything quickly about these? I see some questions in front of me.
We do have one. We've got more questions coming in right now. That's good. People can of course go, but I've given the time. So the important question was that, I just want to emphasize, I'm so glad somebody asked that question. I'm going to say about a year, 18 months after had already engaged change. Okay, so that was the first diagnosis. So that wasn't a question. So that good for your GP. Good for your GP. If that's correct, sometimes patients who have postural tachycardia are diagnosed with MSA. So the other side happens, but it sounds like that GP is very thoughtful. Any idea when this will be available in Canada? I no idea about that. At all. Let me ask you this, right? Depending on, I don't know if she's talking about the webinar or the testing. This test,
I see. I was asking about
might very well be plausible in a hospital like in Toronto, Montreal. Is that possible? I noticed you had Dr. Lang on a few of the papers. Is that something that's going? Yeah, she wants to know if the test is
the test. Yeah, I imagine there are ways of getting it done in Canada through your physician or even I imagine you can, I've contacted the company and asked who the Canadian physicians who have expertise in skin biopsy.
Okay, very good. So let me close it out then. I want to
Larry,
go ahead. Yes.
we're actually going to bring Carol Langer into this. She
okay.
wants to make a quick statement.
Okay, very good. Thank you. All right. All right. Carol, you are on mute.
So I see. Okay, so really quickly, a couple of
Carol,
things.
can I ask you to hold it a second? You know, I just got an email that I have a 2 o'clock patient. So 1 second.
Okay.
Well, I'm going to say something while he's doing that because he doesn't need to hear this. The one thing I was going to say is this really strikes me as a validation of the MSA Coalition Seed Grant Program. You know, just the concept of the Seed Grant Program from the very beginning was to take a good idea and enable it to come to proof of concept so that it can attract funding from bigger fish, you know, because we understand, the MSA Coalition understands that we can't fund to the extent needed to do some of these things. So, you know, from that little tiny seed of that $25,000, we've got a big oak. So this is a good thing. Or I guess that would be an acorn, but anyway, you know, you got the concept. Maple, we had a maple. So that, and then the other point that struck me, and I've been close to this research for a long time, is this is a really amazing place that we are right now. This took 2.5 decades, roughly. So research takes a long time. And for those of us, myself included, who've been frustrated by the lack of progress, it's because research takes a long time. It's not easy. So those are my 2 points I wanted to make.
Yeah, look, you know, those are great points, and you know, I think it's a very exciting time. I mean, I spoke about one very small aspect of the research, and I think we really are positioning ourselves perfectly with what's happening in the pharmaceutical industry, what's happening in basic science labs like the lab that Ariana works in, what happens in the clinical translational space that we work in. And I think to me, you know, I am really looking forward to the next 2 decades of research in this area, and I hope I'm going to be around for all of it.
Well, we all hope that for ourselves and for you. But those are 2 things that struck me as I watched this and listened to this, and I wanted to make sure that everybody was sort of thinking along those things too.
Yeah.
Well, we're about now down to close, and we've had a special guest come in, Carol Langer, who's on the board and is soon to become a retired, I think, board emeritus, and will be helping us further as we move along.
Oh, you know, you can. I'm never going away. I'm just changing my role.
Good. That's good. We also would like to thank Dr. Roy Freeman for taking the time to share what he did. And as you heard, we may hear some more exciting news coming up somewhat soon if the paper gets approved and the review process works. So we might be looking for that, and it would be on our coalition website under treatment pipeline. You'll see a link for that. We also want to thank each and every one of you who donate monies to the MSA Coalition and who have helped us fund the 4 projects we shared this year. If you missed the first 3, or if you want to see this one, as you've already heard, they will be on our YouTube channel. It'll be put up very soon by our executive staff. Prior to this year's annual conference, the 2022 Seed Grant Program awards were made. 7 approved proposals totaled approximately $350,000. Shortly, we will have another group of researchers like Dr. Roy Freeman ready to start research into their hypotheses. From bench to bedside, this is the promise and hope of MSA research. And so we thank all of you. We wish all of you happy holidays. Casey, thank you for helping us man this. Dr. Freeman, the best to you, and the same to you, Carol. Ariana, good luck to you. Thanks, everyone. Bye.